in today's world of COVID, it's pretty interesting to think about what the humoral response is, right? How do we produce antibodies against things like COVID? And this was the entire focus of my energy and my PhD acquisition, right? So I know that we're not all immunologists here, but I'm sure that many of you have gotten your first and or second dose of either a Pfizer or Moderna. But the idea behind a vaccine and a boost is that upon initial introduction to the body, you've got this antibody that's produced, right? So if it recognizes COVID in the body, it'll stick to that spike protein, and then the COVID virus can't get in the rest of your cells and you can clear it. But after that second initial vaccination, you get this dramatic increase in the really protective antibodies. So the question that we were trying to answer in our lab was, what makes a long-lived immune response long-lived, right? So what's really cool is we know from seminal work done back in the early 2000s that vaccination strategies to things like measles, circulating protective antibody titers in the body can last on the order of 3,000 years. So just think about that, right? You have cells that are created, that are making these antibodies that will protect you for 3,000 years, but I don't know anyone that's lived for 3,000 years. So what's special about these cells? And this was a very interesting scientific context and question that we wanted to try to answer. So really it all boiled down to the types of cells that we were evaluating. So, I know that you haven't had a lot of immunology, but we'll focus on this little B cell on the left-hand side. These cells are the cells that when you turn them on, produce those Y-shaped sticky antibodies that'll stick to COVID. Sometimes they'll stick to tumor cells. You can make them stick to anything, right? So we were dealing with the cells that end up creating these antibodies in the bone marrow. And what they do is they get created. So these long-lived plasma cells, the reason you can have these 3,000 years of protection is they go to this special survival niche in the bone marrow where they can live basically for the lifetime of an organism. So we were looking at all the cellular and molecular dynamics that go into this. And really we focused on plasma cells and their transformed or counterpart that's cancer and multiple myeloma. And yeah, there are a lot of complicated pieces to this bone marrow survival niche space, and that was kind of the focus of our work. But what we understood is that upon the differentiation or the transition from one of these B cells that doesn't spit out antibodies and these plasma cells that do spit them out, is there will be a genetic program induced that define the biology, survival function of that plasma cell. And so we found this interesting protein CD28, which is usually expressed on different cells. So we did some basic flow cytometry analysis. And if you're not familiar with this, what you're doing is basically a dot is going to show up if the protein is expressed. So this antigen down here, CD138, is on plasma cells. CD28 was the protein we were looking for. So we evaluated what the long-lived plasma cells looked like in the bone marrow, and they express CD28. They're also expressed on the short-lived plasma cells out of the spleen and other secondary lymphoid organs, whatever. But what's interesting to us at this point scientifically is that, hey, here's a protein that looks like it might be upregulated at a genetic level that no one's ever described before in these cells. Might there be a role, a functional role for this gene or this protein in that plasma cell compartment? So we compared wild type mice to mice that don't have this protein of interest, right? And what was interesting, and when we looked at these long-lived plasma cells in the bone marrow, they were gone in the CD28 knockout mice, right? But they were still present in that short-lived compartment. So it said to us, hey, maybe in these mice, this protein is playing a really important role in keeping these suckers alive. So we did some interesting studies where we vaccinated the animals, and I'm not going to walk you through the entire complicated, vaccination strategy, but what we're looking at, this is what I want you to see on the right-hand side. What these are antigen-specific antibody titers. So if I vaccinated you today to COVID, right, that would look like these black dots over time. And each of these is an individual mouse, right? So yeah, we vaccinate you. Look, your anti-COVID titers go up. For this, it's a NIP-OVA, but it's the same principle. But when we did this in the CD28 knockout mice, look, those antibody titers were not sustained, right? So this was a functional study that kind of confirmed our initial hypotheses. Everything's going well. So what do? We take the cells out, activate that protein, and see if that creates a survival program in the cells, right? So we did out of these mice is we took out the plasma cells, and if you have full serum, right, they're alive. So what we did is we took the serum out in order to uncover a potential survival advantage, right? So with a control antibody, nothing happens. We activate our protein and look, the beautiful scientific result, all the cells survive. Not the case for those short-lived plasma cells. So this all led to a really cool story where we described how CD20 activation in these cells was driving a metabolic program for survival. It took a lot of years. It was a lot of fun. What we continue to understand in the field, right, is that cancer immunotherapy is the future of strategies in cancer, right? So we have, for the past 100 years, used things like radiation and chemo to destroy cancer cells in the body. They target those cells effectively because they are rapidly dividing. What other cells in your body are rapidly dividing? Well, your intestinal lining and your hair. That's why when you have chemo, you get really sick. So what's great about the immune system is not only is it very specific in what it sees, but it remembers. In 2017, the very first cell-based therapies were approved for clinical use. And that got us thinking, well, how do those cells come into the pharmaceutical company? What kinds of cells are being used? And it really started us to think about how we might be able to use an approach that was distinct in the market to bring a solution to a problem. So once again, the immune system, right? We've talked about this. Immune cells are very good at fighting against bacterial and viral infections. But in the capacity of immunotherapy, right, and fighting against cancer, the market is doubling every year from pharmaceutical companies. Whether it's Novartis, Pfizer, Bristol Myers Squibb, everyone that we've spoken to, where they used to be dependent on small molecule inhibitors to a given kinase cascade or signaling cascade inside the cell, right now, everything is biologics based, right? So immunotherapies creating antibodies that either stick to a tumor cell and allow the immune system to eat it up, or cells that you create, put back in the body, right, to fight that cancer. So I like to tell this story, right? This is Emily Whitehead. She was diagnosed with a very aggressive form of leukemia when she was just 5 years old. So at the time, she was at the University of Pennsylvania. And what her doctors did under Carl June and Bruce Levine was they used a new technology where they took out killer cells in her body, the T cells, kind of the tip of the spear when it comes to the immune system's ability to kill other cells. What they did was they introduced a genetic construct, basically a piece of Velcro on that killer cell that could specifically stick to her tumor. So she had one injection of those cells. We call them CAR T cells. Not only is Emily cancer-free almost 10 years later, but she still has those cancer-killing immune cells in her body. So if the cancer ever comes back, her immune system can fight it off. This is the first set of examples of curative strategies in cancer, right? That's an amazing thing to say. We always talk about remission. We always talk about no detectable disease, but never before until recently have we in the field been able to talk about cures. Now, the problem that we saw, and what is clear not only in the data, but the clinical results, is that immunotherapy can work really well in the patients that it works in. So Emily was one of the very lucky few. Unfortunately, 70% of people fail. So we started to ask the question, what's the real problem? Why don't these cells work in the creation of either a cell-based therapy or in order to do the killing part when you add something like an antibody that will stick to a tumor cell? Well, we know that cancer itself turns off the immune system, very well characterized. We also know our immune systems break down as we get older. Not only that, but most of the immunotherapies currently in the market are second line, third line, or even fourth line therapies after chemo and radiation, both of which destroy the immune system. So the problem is evident. We just decided, hey, why don't we just save these cells before you get sick? Is before you get sick, we isolate your cells, we analyze them so that you know what you have is healthy. It can be used for a pharmaceutical design later. We store them, and if you need them, we ship them back to you. So simple idea. All right, this was a year and a half ago. It's gonna be $1 million, right? So what's the go-to-market strategy? My marketing guy at the time said, well, we're going to sell to rich people, right? Of course, that makes sense. It's not quite how business works. So this began the new journey of understanding that, hey, we built this great solution. Now, all we need is to know who really has that problem and how we can address it with what we've created, right? So this is the opposite of what you want to do in business. And this is often what we see in the laboratory is we'll squirt liquid in tubes and we'll find something that works great as a technique or a diagnostic. And then we try to roll it out and we're like, everybody's going to want this. But in science, oftentimes we bring something to market that frankly, nobody wants. A lot of the side effects are due to the variability of the immune cell subsets that we use for a given clinical application. So when you look at something like a CAR T cell, the kinds of cells that Emily got, it's neurotoxicity, it's cytokine release syndrome, which is basically a storm of inflammatory proteins that happen in the body when you put the cells in. And it's kind of this balance, it's a dance, because when you have toxicity, it means that the cells are killing off the tumor. So we use things like steroids, which turn down the immune system. We use antibodies that block some of those pro-inflammatory signals. But what our approach will allow us to do from a larger clinical approach or execution of the approach is to have a standardized set of biological material that goes into the development process. So when you look at the percentage of patients that undergo either a monoclonal antibody regimen or a cell-based regimen, that comprises about 60% of the total biologics and gene therapy umbrella in pharmaceuticals. For us, the genetics component is a little bit less. Our cells may not help with that. Any therapy that you would use that depends on an immune system for the anti-tumor function, for the actual killing, that is what you can use your healthier cells for. But imagine what we could do tomorrow. If you have a pool, let's say you're 65 years old, and you've got your 40-year-old immune system stored down, and global pandemic #2 hits. And we're waiting for that vaccine. We're waiting for that vaccine, right? And you've got billions of immune cells available. You can just go into your doctor's office, get a biweekly injection of your healthier immune soldiers, right? And that may be able to protect you from developing that disease to the point at which you're intubated and potentially pass. What we know from 3 decades of umbilical cord blood storage is that they're always healthy, right? Our cryopreservation methods today, you put them into liquid nitrogen, those cells are in stasis. There may be a very small loss of their survival over time, but the beauty about immune cells, especially and specifically the T cells, which are the killer ones in the body, and the B cells, which create those antibodies, is they undergo this natural turnover called homeostatic proliferation. So that's just fancy science term for if I don't feel great in the body, I'm just going to create a genetically identical copy of myself. That's how you can get vaccinated today against measles, but those cells stick around long enough to provide protection 3,000 years down the line, right? That one cell doesn't necessarily just park it and stay forever. They have to go through that turnover because cellular fidelity, like the lifetime of an individual given immune cell, is highly variable, but that's why you want to keep what your immune system knows so that you can maintain that memory. And in order to do so, you have that way of natural turnover. So if I give you your cells tomorrow that were stored 10 years ago and the cells aren't happy being woken up, and I'm sure if you've been in a lab where you've pulled cells out of the tank, they're not always that happy, put them back into the body, they will go through that genetic turnover and you still have that full complement of your soldiers that were healthier 20 years ago or 10 years ago than they were the day that you want them to use.